In this lesson, we will learn about signal transduction in animals. Conversation in biological world is quite natural. Even on the level of the cell, a busy broadcast of communication is occurring. Signal transduction is the study of how a cell communicates. Every cell is able to communicate through having evolved the ability to produce, recognize, interpret and respond to signals in its environment. The word signal in this context refers to nothing more than chemical molecules that are floating around. Cells have learned to detect many of these chemicals. Their molecular detection components produced by the genes they contain allow the cell to converse in this chemical language of the cell. When you come right down to it, this ability to communicate has allowed the cell to evolve. If a cell could not receive or respond to signals from its environment, for example, sensing food or predators, it would be unable to adapt its behavior and over a time would be out-competed by those that could communicate. Therefore, it does form a vital part of a cell. Where did the term signal transduction originate from? The signal transduction is an umbrella term in biology. It is used to refer to a broad area of cellular biology research involving topics such as chemical signals used by cells, how these signals are received, how a cell interprets them, and the ways in which cellular machinery can be used to respond. Essentially, signal transduction ensures that a message can be converted from one form to another during its travels and still retain its original content. Let's look at human communication as an illustration. Consider how a message is sent over a telephone. One person speaks into a receiver that converts the sound into an electrical signal, which can then be transmitted over a great distance before being converted back into sound at its destination. This process retains the original content of the message and is called signal transduction. The signals sent by cells are far simpler than the highly complex messages used by humans. One cell, termed the signaling cell, produces a particular chemical molecule that is detected by another cell. The receiving cell, using a receptor protein that recognizes the molecule and responds specifically to it. The protein, acting as a receptor, is the first step in which the chemical signal present on the outside of the cell will be converted, that is, transduced to different signals inside the cell. These signals will subsequently direct cell behavior. Multicellular organisms have developed a variety of mechanisms allowing very efficient and controlled cell-to-cell communication. Depending on the distance that the signaling molecule has to travel, we can talk about different types of signaling. The first one is endocrine signaling. Here, hormones are produced by an endocrine gland and sent through the bloodstream to distant cells. Second one is paracrine signaling. Here, the signaling molecule affects only the target cell in the proximity of the signaling cell. An example is the conduction of an electrical signal from one nerve cell to another or to a muscle cell. The third one is autocrine signaling. In autocrine signaling, cells respond to molecules they produce themselves. Examples include many growth factors. Fourth is signaling by the plasma membrane attached protein. The proteins attached to the plasma membrane of one cell could interact directly with receptors to an adjacent cell. Fifth one is neuroendocrine signaling. The neurosecretory neurons of hypothalamus produce neurosecretory hormone, the releasing factors which act as signaling molecule for pituitary, which secretes its own hormones. And the last one is signaling via gap junctions. Gap junctions are specialized cell-cell junctions formed between closely opposed plasma membranes and which directly connect the cytoplasms of the cell via narrow channels. These channels allow the exchange of small intracellular signaling molecules or mediators such as calcium and cyclic AMP, but not macromolecules. Hence, these cells can communicate with each other via gap junctions. The list of signals that have been discovered numbers in the hundreds and grow longer every day. Some examples include protein, peptides, amino acid, nucleotides, steroid and gases. Molecular signals sent by one cell is processed and converted into response by series of steps via signal transduction. Most signaling molecules, for an example protein hormones targeted to a cell, bind at the cell surface receptors embedded in the plasma membrane. Signaling molecules that are able to cross the plasma membrane, for an example steroid hormones, interact with the intracellular receptors. Hence, according to the nature of signaling molecule, there are two different pathways for signal transduction. They are signal transduction for protein hormones and signal transduction for steroid hormones. Other signaling molecules are neurotransmitters, growth factors, cytokines, nitric oxide and dopamine. Now we will study signal transduction for protein hormones. In 1971, Martin Rodbell and his co-workers, while working on rat liver cells, found that a GTP binding regulatory protein called G protein activates adenylate cyclase. Thus, Sutherland's cascade phenomena is modified. A protein hormone secreted by an endocrine gland travels via the blood stream to deliver a signal to a target cell. Small molecule, peptide and protein hormones are hydrophilic and cannot cross the membrane barrier of the target cell. So, hormone binding to transmembrane protein receptors transduces the signal across the cell membrane that produces a cellular response. The hormone membrane receptor is a large protein that consists of 7 hydrophobic transmembrane spanning regions. The free amino terminus of the receptor protein is external to the cell and the free carboxyl terminus is internal. The receptor is associated with G protein. G protein receptor comprises of alpha, beta and gamma subunits. The peptide hormone assumes its active configuration as it approaches the extracellular active site of the receptor, thereby locking into the active site and transferring its information. The information transferred to the interior of the cell result in a mechanical interaction between the receptor and a G protein. Mechanically, activation of G protein result in a transfer of GTP for GDP on the alpha subunit. The GTP activates the alpha subunit, which separates from the beta and gamma subunits and interacts with an inactive adenylate cyclase within the plasma membrane, stimulating the synthesis of cyclic AMP, that is second messenger. Cyclic AMP activates protein kinases A. Kinases are enzymes that transfer a phosphate from ATP to other proteins, that is to bring about phosphorylation. The alpha subunit is a slow GTPase and thus the GTP is hydrolyzed to GDP after a period of time. The alpha subunit then reassociated with the beta and gamma subunits and adenylene cyclase is inactivated. The protein kinases A has 2 regulatory subunits and 2 catalytic subunits. Attachment of regulatory subunit to catalytic portion makes protein kinases A inactive. When cyclic AMP binds to regulatory subunit, the tetramer releases its 2 active subunits. Active catalytic subunits pick up energy from ATP and then act on enzyme in the cell, that is, it phosphorylase and activates it. Glycogen phosphorylase is responsible for glycogenolysis, the breakdown of glycogen to glucose. Protein kinase A initiate a signal transduction cascade that result in the intracellular breakdown of glycogen to glucose. In case of adrenaline as a hormone, the net result is increased muscle energy. Thus, activation of surface receptors switches on the activity of other intracellular signaling proteins. In turn, these proteins activate other proteins in a series of events called as signal cascade. Because of this cascade, signal transduction is very useful in conducting a fast response. In each step, the original hormone, that is signal, is amplified, and since the method uses the little amount of hormone to create a large effect, the body does not need to waste time and energy producing a lot of that hormone. Now let's discuss about what second messenger actually is. Wilbur Sutherland Jr. discovered second messengers, for which he won the Nobel Prize in Physiology in 1971. Second messengers are small molecules or ions such as nucleotide or lipid derivatives, which elicit changes in a metabolic pathway some distance away from the membrane. They greatly amplify the strength of the signals. For an example, cyclic AMP, calcium, lipids, ionositol triphosphate, diacylglycerol, nitric oxide, and free radicals. Secondary messenger systems can be synthesized and activated by enzymes like cyclases that synthesize cyclic nucleotides, or by opening of ion channels to allow influx of metal ions like calcium signaling. These small molecules bind and activate protein kinases, ion channels, and other proteins, thus continuing the signaling cascade. Secondary messengers are a component of signal transduction cascade. Their production and destruction can be localized, enabling the cell to limit space and time of signaling activity. Now let's see what G proteins are. G proteins, that is guanine nucleotide binding proteins, are a family of proteins involved in transmitting chemical signals outside the cell and causing changes inside the cell. They communicate signal from many hormones, neurotransmitters, and other signaling factors. G protein is a heterotrimer. It has got 3 subunits: alpha, beta, and gamma subunits. Beta and gamma are alike and occur as dimers. Most important is alpha subunit, which is different from others. All G proteins except GP and RAS proteins are heterotrimer. It is GTP binding regulatory protein or guanine nucleotide binding regulatory protein, which may be of inhibitory or stimulatory type. They are widely distributed and unique to eukaryotic cells and are involved in the responses produced by functionally different numerous receptor types such as ion channels in the nerve and muscle cell, hormone receptor in all types of cells, and photon receptors in the visual system. Sutherland and Martin Rodbell have described the mechanism of action of protein hormones. Rodbell's important discovery of G proteins and cell signaling has paved the way understanding the occurrence of several disorders associated with defects either in the G protein itself or in its receptor. These are pituitary thyroid tumors, combined precocious puberty, pseudo-hypoparathyroidism, McQueen-Albright syndrome caused due to defective G protein, that is GS-alpha, and adrenocortical ovarian tumors caused due to defective G protein GI-alpha. Further exploring human diseases caused due to defective G protein receptor. First one is hyperthyroidism. It is due to defective G protein receptor, that is TSH receptor. Second is retinitis pigmentosa, which is due to defective rhodopsin receptor. Third one is familial glucocorticoid deficiency, which is caused due to defective ACTH receptor. Moving ahead to recycling of protein hormone receptor. Most of the protein hormones bind to membrane receptors to elicit their actions. After binding, the hormone receptor complex is metabolized. This process helps to terminate the signal, leading to bring about hormone action. Receptor with ligand aggregate, and after aggregation, the pits bud off, that is endocytosis. After endocytosis, it quickly loses its clathrin coat. Clathrin coat is important only for endocytosis and form curl, that is compartment for uncoupling receptor and ligand, that is receptosome. Receptosome acidification occurs using ATP molecule, and due to this there is separation of ligand and receptor in such a way that ligands remains in the vesicle and receptor remains in the extended tubule. Extended tubules are able to move towards Golgi apparatus and there they fuse with Golgi. Modification of receptor occur in Golgi apparatus and then this receptor is moved from Golgi in the form of vesicle to plasma membrane, and by exocytosis this receptor remains on the plasma membrane. In this way receptor is ready to receive another ligand, and ligand fuse with second lysosome, where lysosomal enzyme break the ligand and makes it inactive and exocytosis occurs. In this way protein hormone receptor is recycled. Now we will study signal transduction for steroid hormones. Steroid hormones are essential regulators of key physiological processes such as reproduction, glucose metabolism and the response to stress and salt balance. The biological effect of steroid hormones are transduced by intracellular receptors that directly mediate the action of their cognate hormone. Unlike protein hormone, however, most hydrophobic signaling molecules can diffuse across the plasma membrane and bind to receptors inside the target cell, in the cytoplasm or nucleus. Steroid hormones are one such example. Steroid hormones are smaller and hydrophobic molecules that binds to intracellular receptors in the target tissue to elicit its action. The steroid hormones are C18, C19 and C21 compounds produced by the gonads, adrenal cortex and placenta. Mechanism of steroid hormone action. As soon as the steroid enters the cell and binds to the receptor component of the receptor protein complex, causing dissociation, release of heat shock proteins and activation of receptor. The activated receptor develops an affinity for DNA and is translocated from cytosol to nucleus, where it binds to DNA segments. The DNA binding results in a change in the transcriptional rate of specific genes. The resulting production of more or less mRNA under the control of transcription factors and RNA polymerase leads to the changes in the content of specific proteins in the target cell. Properties of nuclear receptor. Receptor for steroid hormones are known as nuclear receptor as they are located inside the nucleus of the target cell. Receptors located in nucleus are active and small in size, while those located in cytoplasm are inactive and large in size. All are proteinaceous in nature and are heat sensitive. They are present in cytoplasm in the form of homodimers attached with heat shock protein, that is, steroid receptors exist intracellularly as homodimers with a non-steroid binding HSP. The nuclear receptor would recognize the signal, that is steroid hormone, and transduce its biological effect. The receptors belong to superfamily proteins, all of which are regulators of gene transcription. The superfamily consists of the receptor for steroid hormones, thyroid hormones, vitamin D3 and retinoic acid. Members of this family share three important properties. They each bind a hormone ligand and further bind to specific DNA sequence, and thereby they act to modify the transcription activity of specific genes. All steroid receptor has similarity and are considered as prototype receptor. It has three domains, which are amino-terminal domain, central domain, that is DNA binding domain, carboxyl terminal end region or ligand binding domain. Now let's see recycling of steroid hormone receptor. The fate of steroid receptor complexes after their nuclear retention in the target cell is not firmly established. Nuclear glucocorticoid and androgen receptor complexes could be recycled back to the cytosol in their responsive tissues, whereas this has not been clearly established for the case of progesterone and estrogen receptors. The models of steroid receptor recycling proposed so far involve release of chromatin-bound complexes into the cytosol, loss of steroid, and receptor inactivation. These receptors, however, can eventually be reactivated to a steroid binding form to reinitiate a cycle of steroid binding and further nuclear translocation. We propose that this model can represent a general aspect of steroid hormone action, provided that inactivation or reactivation processes occur in every steroid responsive system. A process involve a reversible receptor inactivation could play a major role in the control of steroid receptor recycling. It is proposed that a control on the extent of receptor available to steroid binding could result in a modulation of cellular responses to steroid hormones.
At the end, to conclude, this lesson described process of cell signaling, types of cell signaling found in different cells, signal transduction and their importance. It is further followed by signal transduction for protein hormones. Further, the lesson explores note on second messenger and G proteins. Rodbell's important discovery of G proteins and cell signaling has paved the way in understanding the occurrence of several disorders associated with defects either in G protein itself or in its receptor has also been discussed. Lastly, signal transduction for steroid hormones and note on nuclear receptor and its recycling has been mentioned. Thus, in this manner, complete signal transduction in animals has been studied.